Good morning, guys. Today I want to talk about renal physiology. In the renal physiology and making urine, we have three important steps. One of them is called glomerular filtration. One of them is called tubular reabsorption, and the other one is called tubular secretion. And in this part, first I want to talk about glomerular filtration. As you see in this picture, you have the glomerulus here and the glomerular capsule or Bowman's capsule. The filtration take place in this part. The filtration means that moving substances from the blood vessel to the Bowman's capsule. And when these filtrates start to flow in the renal tubule, we have two other processes. One of them is called reabsorption, when the essential material in the nephron come back again to the blood vessels, and the other process is called tubular secretion, which we have some waste products, some drugs, excess ions, which move from the peritubular capillary and vasa recta to our renal tubule. And in this part, I want to talk about these three processes in our kidneys. The glomerular filtration is the first step. The filtration uses the pressure to force substances across the capillary wall. So we have blood pressure here, and you know that we have the opposite pressure, which we call it capsular hydrostatic pressure and blood colloid osmotic pressure, which I will talk about them later in this part for you. The water and the small molecules move out of the glomerulus. During each day, 150 to 180 liters of water pass through the glomerulus to the Bowman's capsule. This is the meaning of glomerular filtration. The next step is called tubular reabsorption. As the filtered fluid flows along the renal tubule and the collecting duct, tubule cells reabsorb about 99% of filtered water and many useful solutes. The water and solutes return to the blood, flowing through the peritubular capillary and the vasa recta, that capillary network which wrap around our nephron. Pay attention. The tubular reabsorption returns substances to the bloodstream. In contrast to the absorption, which means entry of new substance into the body, which occur in our GI tract or in our digestive system. And the next step is called tubular secretion. As the filtered fluid flows through the renal tubule and collecting duct, the renal tubule and collecting duct cells secrete the other material, such as waste products, drugs, and excess ions, and also urea, uric acid, and creatinine to the renal tubule. So the renal secretion means that removing of substance from the blood and bringing them to the renal tubules. And at the end, the urine volume is the amount of filtration and secretion minus the amount of reabsorption. The glomerular filtration. The fluid that enters the capsular space is called glomerular filtrate. On average, during the day, the volume of glomerular filtration in adult is 150 in female and 180 liters in male. More than 99% of this glomerular filtrate should return back to our bloodstream by tubular reabsorption. And at the end, we only have 2 liters of urine which is produced during the day. And the filtration fraction is the fraction of blood plasma in the afferent arteriole which enter to the nephron. We call it filtration fraction. In this picture, you can see the filtration membrane. I talked about this filtration membrane before for you. In the first part, we have the fenestrated capillary. The fenestrated capillary is capillary type 2. We have some pores or fenestration in the endothelial cells. but we have the complete basement membrane around this capillary. And after that, we have the visceral layer of Bowman's capsule. The cells in the visceral layer are called podocytes. They are simple squamous epithelial cells which contain many feed processes, and their feed processes are called pedicle. These feed processes go interdigitally into each other, and between these pedicles you can see some slits. In this picture you can see the parietal layer and also the proximal convoluted tubule with its microvilli and brush border too. In this picture, you can see the filtration membrane from the side. Here, you can see the fenestrated capillary, and in this part, you have the fenestration or pores. After that, we have a complete basement membrane, and then these yellow structures are the podocyte. Between these the pedicle of the podocyte, and between these pedicles, you can see the slits. So some material can easily pass through these pores and go to the Bowman's capsule if their sizes let them. The other thing which is important for this movement is the charge of the material. You know that the charge of the basement membrane is negative. So positive and neutral particles can easily pass, but negative particles are repelled by this basement membrane. And the proteins, the red blood cells, white blood cells, and platelets are bigger than these pores, so they cannot pass through this membrane and they cannot come to the nephron. You need to pay attention that if we see any protein in the urine, if we can detect any red blood cell, white blood cells in the urine, it means that we have a problem in our kidney, like some kidney failure. So the presence of this material is unusual and abnormal in the urine, and this is the function of this filtration membrane, which lets some material to pass and it doesn't let the other material to pass easily. The process of filtration is the same in glomerular capillaries like the blood capillary which we have in all parts of the body. However, the volume of fluid filtered by our renal corpuscle is much more larger than the other capillaries. Last time I told you that each time between 20 to 25% of our resting cardiac output are in our two kidneys. So we have more filtration in the glomerular capillary than the blood capillaries. We have three reasons for having more filtration in glomerular capillary. The first one is this: the glomerular capillaries present a large surface area for filtration because they are long and extensive. Between these capillary network, we have some mesangial cells which can regulate how much of this surface area is available for filtration. When mesangial cells become relaxed, the surface area is maximal, and the glomerular filtration is very high. When they go to contraction, they reduce the available surface area and the glomerular filtration decrease. The other reason is this: the filtration membrane is thin and porous, despite having several layers, but the thickness of this filtration membrane is only 0.1 micrometer. So it's only 0.1 micrometer in its thickness. The glomerular capillaries also are about 50 times leakier than the capillaries in the other part of our body because they have large fenestration, and their fenestration doesn't have any valve to protect them and cover them. And the last reason is this: the glomerular capillary blood pressure is very high. The efferent arteriole is smaller in diameter than the afferent arteriole, and it makes a resistance to the outflow of the blood from the glomerulus. As a result, blood pressure in glomerular capillary is very higher than the other capillaries in our body, so filtration can take place more and more. In this picture, you can see the different pressures which we have in this renal corpuscle. The first pressure, you know about all of this pressure from the blood vessel chapter, but I want to talk about all of these pressures for you. The first pressure is called glomerular blood hydrostatic pressure. The glomerular blood hydrostatic pressure is the blood pressure in the glomerulus. It promotes filtration by forcing water and solutes in blood plasma through the filtration membrane. The amount of this pressure is about 55 millimeters of mercury. The other pressure is called blood colloid osmotic pressure. This blood colloid osmotic pressure is due to the presence of protein in the blood plasma, like albumin, globulin, and fibrinogen. The blood colloid osmotic pressure opposes filtration by drawing water from the filtered back into the plasma within the glomerulus. I talked about this pressure before for you. The albumin is very similar to basil seed or chia seed. They like to reabsorb water. They like to have a coat of water around themselves. So they make pressure on the water and bring them near themselves. The next pressure which we have is called Bowman's capsule hydrostatic pressure, or the other name is capsular hydrostatic pressure. Is the hydrostatic pressure exerted against the filtration membrane by the fluid already in the Bowman's capsule. Capsular hydrostatic pressure is opposite to the glomerular hydrostatic pressure, and it helps the material to go out of the Bowman's capsule and come back again to the glomerulus. The amount of this pressure is about 15 millimeters of mercury. And the last pressure, which is zero in our nephron and in our kidney, is called Bowman's capsule osmotic pressure, because in the Bowman's capsule we don't have any protein to help making pressure on the blood and bring the water and bring the water near themselves. So the amount of this pressure is zero in our renal corpuscle. And the net filtration rate is this. The fluid moves from the glomerulus into the capsules. And I will show it to you in the next slide. Here you can see the glomerular hydrostatic pressure, or the blood hydrostatic pressure, which is 55 millimeters of mercury, and you can see it by the purple arrow. The other pressure is the blood colloid osmotic pressure, the pressure of the protein in the blood. It's about 30 millimeters of mercury, and you can see it by this red arrow. And the last pressure which we have is capsular hydrostatic pressure. It's about 15 millimeters of mercury, and the result of all of these pressures is 10 millimeters of mercury. By the pressure of 10, all the materials can come out of the blood capillary and come to the Bowman's capsule. Not all the materials which can easily pass. And if I want to write the equation here for you, I can write it like this. The net filtration pressure is equal to 55 which is the glomerular hydrostatic pressure, minus 30 plus 15. And this result is equal to 10 millimeter of mercury. This is the net filtration pressure. You can check this animation later for yourself. It talks about the renal filtration. The amount of filtrate formed each minute in all of the renal corpuscle of our both kidneys is called glomerular filtration rate, or GFR. In adult, the GFR is between 125 milliliter per each minute in male and 105 milliliter per each minute in female. The homeostasis of our body fluid requires that the kidneys maintain the relatively constant GFR. If the GFR is too high, needed substance may pass quickly and they cannot return back to our blood vessels. And if the GFR is too low, nearly all of the filtrate may be reabsorbed and some of waste product, toxins, and the material which we don't need them come back again to the blood vessels. So for having homeostasis, we need to maintain our GFR in a constant level. And as I told you, the GFR is 125 milliliter per each minute in male and 105 milliliter per each minute in female. The GFR is directly related to the blood pressure that influences the net filtration pressure. Any changes which we have in the blood pressure can affect our GFR. If the blood pressure drops, the GFR also decrease. The glomerular filtration decrease altogether if glomerular blood hydrostatic pressure drops to 45 millimeter of mercury. And if the blood pressure goes up, the GFR increase too. The glomerular filtration rate is controlled by three different mechanisms. One of them is called renal autoregulation, one of them is called neural regulation, and the other one is called hormonal regulation. And all of these three mechanisms work together in two ways. First, they want to adjust the blood flow into and out of the glomerulus, and then they want to change and alter the glomerular capillary surface area available for filtration. GFR increases when blood flow into the glomerular capillary increase, and GFR decrease when the amount of blood which come to the glomerular capillary decrease. If you look at this picture, you can see the afferent arteriole, the glomerulus in the center, and the efferent arteriole in the other side. And filtration takes place in the glomerulus. Now look at the picture in the left side. In this picture, you see that the afferent arteriole dilates, and we have constriction of efferent arteriole. In these cases, the amount of blood which come to the glomerulus increase, and the blood leave here for longer time because our efferent arteriole is very narrow and our blood needs more time to go out of the glomerulus. So when the blood remain in the glomerulus, it can do more and more filtration. And as you see here, the GFR increase. In the other situation, sometimes we have afferent arteriole constriction and efferent arteriole dilation. In these cases, the amount of blood which come to our glomerulus decrease, and the amount of blood which can leave our glomerulus increase. So when the blood come to the glomerulus, it can easily go out, and the amount of GFR decrease. So our body can afferent and efferent arteriole, it can work with dilation and constriction of these two arterioles to change the amount of GFR. Now, I want to talk about renal autoregulation for homeostasis. The kidneys themselves help maintain a constant renal blood flow and GFR despite all the fluctuations which we have in the blood pressure during the day. This capability is called renal autoregulation and consists of two mechanisms. We have two mechanisms here. One of them is called myogenic mechanism, occur when stretching triggers the receptors which we have in the wall of the blood vessels, and the other one is called Tubuloglomerular feedback. And I will talk about these two processes in the next slide for you. In this picture, I want to talk about myogenic response. Here you can see the afferent arteriole, here you can see the glomerular capillary, and this is the efferent arteriole. When the blood pressure goes up, the elevated blood pressure can make a stretch in the wall of capillary. When we have a stretch in the wall of capillary, the response is going to constriction, as you see here. The afferent arteriole go to constriction and the amount of blood which come to glomerular capillary decrease. In the previous pictures, I told you that when the blood pressure goes up, the glomerular filtration rate increase. By this mechanism, by this myogenic response, our body do the opposite function because it wants to keep homeostasis for us. And in the next picture, you can see the drop in the blood pressure. When the blood pressure drops, our GFR should decrease too. But at this time, when we don't have any pressure on the smooth muscles in the wall of the afferent arteriole, the afferent arteriole go to relaxation, and so we have more blood which come here and our GFR increase. We call it myogenic response. And the myogenic mechanism occurs when stretching triggers constriction in the wall of our blood vessels. Specifically, our afferent arteriole is affected in this mechanism. And the other mechanism is called tubuloglomerular feedback. The tubuloglomerular feedback is the mechanism which happens in the macula densa. I hope you remember what is the macula densa. If you remember from kidney anatomy, I told you that when the distal convoluted tubule and the afferent arteriole come near each other and attach to each other, we have the apparatus which we call it JG apparatus or juxtaglomerular apparatus. These macula and the cells which we have in the wall of distal convoluted tubule is called macula densa. It can provide a feedback to our glomerulus. Now I want to talk about it. When the GFR is above normal due to elevated systemic blood pressure, the amount of filtered fluid which move in the renal tubule increase. As a result, the proximal convoluted tubule and the nephron have less time to do reabsorption of sodium ion, chloride ion, and water. When these filtrates reach the distal convoluted tubule, the macula densa can detect the increased delivery of sodium, chloride, and water. And because of that, it starts to inhibit secretion of nitric oxide from the cells in juxtaglomerular apparatus. When nitric oxide is released, you need to know that it stimulates the afferent arteriole to dilate. When nitric oxide release is inhibited, the afferent arteriole constricts and resulting in decreased blood flow into the glomerular capillaries and decrease the GFR. On the other hand, as the blood pressure falls, filtered fluid flows more slowly along the renal tubule and they have enough time to do reabsorption. So our macula densa cannot detect excess sodium ion, chloride ion, and water, and we don't have any inhibition for secretion of nitric oxide. So the nitric oxide increase. When the level of nitric oxide increase, afferent arteriole dilates, increasing blood flow into the glomerulus and increasing the GFR. Macula densa cells also inhibit release of renin from juxtaglomerular cell when increased sodium ion and chloride is detected in filtrate fluid. And as you know from the blood vessel chapter, this links renal autoregulation of GFR with hormonal regulation of tubular reabsorption and secretion. And I will talk about it later for you. So this is the tubuloglomerular feedback. In this feedback, you see that when the GFR increase, when the blood pressure increase, our macula densa inhibits secretion of nitric oxide, and by inhibition of this secretion, then the blood pressure decrease. And when the blood pressure decrease, our GFR come down. So by myogenic response and tubuloglomerular feedback, our kidneys can control the GFR. The neural regulation is the other mechanism which our kidney can regulate the GFR. Like most blood vessels of our body, those of the kidneys are supplied by sympathetic neuron of the autonomic nervous system, which release epinephrine and norepinephrine. Epinephrine and norepinephrine produce a vasoconstriction of the afferent arteriole. At rest, sympathetic stimulation is low, afferent arteriole become dilated, and renal autoregulation of GFR prevails. With greater sympathetic stimulation, as occurs during exercise or hemorrhage, afferent arteriole go to constriction. As a result, blood can flow into the glomerular capillary decrease, and the glomerular filtration decrease, and the GFR drops. So our blood can go to the other part of our body which we need them. For example, at the time of exercise, we need blood for our muscles. So the blood can easily go to the muscles and supply our muscles. This is one of the other regulation which we have for filtration, and the other one is called hormonal regulation of GFR. Two hormones contribute to regulation of GFR. One of them is angiotensin II. Angiotensin II can do vasoconstriction of afferent and efferent arteriole and reduce the renal blood flow, so it can decrease our GFR. As the blood volume increases, the atria of our heart can secrete atrial natriuretic peptide. The atrial natriuretic peptide have their own receptors on the mesangial cells. They have their receptors on their mesangial cells, and so the mesangial cells become relaxed and they increase the capillary surface area and they can increase our GFR. Atrial natriuretic peptide is secreted in response to the stretch of the cardiac atria. This part is about the filtration. In the next video, I will talk about the tubular reabsorption and tubular secretion for you.